Hello, everyone. Thank you for joining us today and welcome to today's webinar. My name is Nela Halterman and I'm an assistant science editor for The Scientist. I will be moderating our discussion today. Our speaker, Dr. Victor Yin, will discuss how mass photometry and charge detection mass spectrometry enable the study of several challenging protein systems, including the interaction of full antibodies with SARS-CoV-2. We really like for our webinars to be as interactive as possible, so we encourage you to send us your questions or comments at any point during the webinar, and Dr. Yin will address these during the Q&A session following the presentations. If you would like to ask a question, simply click on the Q&A tab on the right side of your screen and type your query into the question box. This webinar is recorded and we will archive it on the Scientist website and send you a link to the recording via email within a couple of days. Please do note that you will not be able to download the presentation slides. Before we start, I would like to take the opportunity to thank our webinar sponsor. As the pioneer of mass photometry, Refine produces a disruptive new generation of analytical instruments that measure the mass of individual molecules directly in solution. Mass photometry transforms the ability to characterize the composition, structure, and dynamics of biomolecules, revealing the true behavior of molecules in their native environment. Refine aims to accelerate research and enable discoveries by sharing mass photometry with the global scientific community. Our sponsor has provided us with some helpful resources related to today's webinar, and we have posted these in our handouts section for you to download. And you can find that handout section similarly on the right side of your screen. You can access and download these documents at any time during the webinar. And with that, let me introduce our speaker. In 2020, Dr. Victor Yin received his PhD in physical and analytical chemistry from Western University in London, Canada. His graduate work was done in the laboratory of Lars Connerman, where he characterized the effects of heterogeneous oxidative post-translational modifications on protein structure, stability, and enzymatic function using an ensemble of mass spectrometry approaches. Dr. Yin is currently a postdoctoral researcher in the lab of Albert Heck at Utrecht University. His research focuses on characterizing viral proteins and large protein complexes with mass measurement approaches, particularly single particle methods. Before we start, I just want to make sure that your slides are up and running. That looks great on my end. So with that, Dr. Yin, please take it away.
Yeah, hi everyone. Thanks for the introduction, and thanks again for the organizers and Refine for helping to set this up. So today I'm excited to tell all of you a little bit about the work going on here in Utrecht, where we're using single particle mass methods to sort of study these heterogeneous complex protein assemblies that have historically been quite challenging. So before I dive into it, I think it's quite useful to first sort of discuss, well, why would we want to measure the mass of biomolecules? One sort of reason why you would want to do this is for biomolecular identification. For example, with proteins, because each protein has a unique amino acid sequence, they'll also have unique masses. So if you were to measure the mass of, say, a mixture of proteins, here two proteins, A and B, you could distinguish them based off just their mass alone, and you don't really need to add any sort of special label or a special tag to distinguish these species. Secondly, measuring mass is actually quite convenient when you want to measure protein interactions. Because if a complex were to form between, let's say, a protein and a ligand, or a protein and another protein, if they form a complex, you would then expect the mass to be the sum of the component masses. So on the right here, if A and B were, for example, interaction partners, we would actually see in their masses peak corresponding to A and B complex. And we could even, for example, distinguish between different stoichiometries. For example, this 1 plus 2b complex, which would be actually a higher mass than the first complex. So there's a lot of reasons why one would want to measure the mass of biomolecules. So because molecules are quite small, you generally need quite specialized instrumentation and quite advanced techniques to measure them. Over here, we're kind of specialists in the probably the most common way to measure these things, which is an analytical technique called mass spectrometry. And basically, mass spectrometry, we take advantage of ion motion, and we use this ion motion to determine the mass of proteins and their complexes. Basically, because we know quite well the physics behind, for example, how an ion will move in an electromagnetic field, we can measure things such as the oscillation frequency of an ion. Here on the right is kind of the type of mass spectrometer that we use, called an orbitrap. We can sort of measure the frequency that an ion moves in an electric field, and that sort of lets us calculate the mass. What's important is that in mass spectrometry, we usually don't directly measure the mass, but because we're dealing with ions, we actually normally measure what's called this mass-to-charge ratio, or M over Z. And the right-hand side here, I just show some standard spectra of various proteins. You'll note that for each protein, we generally see more than one peak in the mass spectrum, and each of these peaks correspond to the same mass, but a different charge value, actually. So an important part of mass spectrometry, in order to really calculate the true mass of our analyte, we need to do something called charge assignment. And in that, we need to figure out actually which charge belongs to which peak. Here, they're all labeled. So there's a number of methods that one can do to go about doing that. For example, you can notice that the higher the charge on the analyte, the more closely spaced these series of peaks are. So the take-home message from this that I want to stick is that to do mass spectrometry, you really need to get a correct assignment of your charges in order to get the correct mass. So there's a few ways of doing mass spectrometry experiments, but the way that I'll talk about today is designed in such a way that things such as protein complexes remain undisturbed, and this approach is called native mass spectrometry. Now, native mass spectrometry has been around as an analytical technique for a few decades now, but historically there's been some known challenges in the field. One of these challenges is the size of your analyte. So when an analyte, so a protein complex of interest, for example, becomes larger and larger, they become harder and harder to manipulate in the ways that we need them to, and actually measuring their ion motion becomes more and more challenging. So historically speaking, our lab has been on the cutting edge of this area, working closely with mass spectrometer manufacturers to design better instruments that can handle larger and larger particles. Secondly, a big problem with native mass, well, not a problem, a major challenge for native mass spectrometry is this concept of heterogeneity. So to illustrate this here on the right, are mass spectra of viral capsid, actually. So this is the hepatitis B virus capsid. And on the bottom here, you can see what the mass spectrum looks like for the capsid alone. So although the spectra look quite complex, you can still see the structure that corresponds to each of the individual charge states. So the mass measurements actually still work quite well under these conditions. However, as we move up on this plot, what was done here is that an antibody fragment was titrated against the capsid. And from symmetry arguments, we would expect to be up to 60 copies of this antibody fragment binding to the virus. And what you see here is essentially a titration curve of this antibody fragment. And what we see is that as a function of antibody concentration, the spectra get more and more poorly resolved. And that's because within this sample, there's many different capsids that have many different numbers of antibodies. So the number of species in this mixture becomes greater and greater. It eventually gets to the point where there's such a high heterogeneity in the same spectral region that we begin to lose the resolution that we had originally for the charge states. And although here, for example, in trace E, we can clearly see analyte, because of the poor resolution in the charge states, it's actually quite challenging, if not impossible, to correctly determine the charge. As a consequence, correctly determine the mass. Here, we were a bit lucky in that these virus capsids are quite close to perfect spheres. So we can make some educated guesses about how many charges they should have. But the fact remains that correct charges assignment in these systems remains quite challenging. And such an approach is not really applicable to all systems. So heterogeneity is quite a large problem in these native mass spectrometry measurements. So today I wanted to talk about two stories that were recently published by our group that are really on the sort of edge in terms of challenging systems. So these systems have historically been quite challenging, but today I kind of want to talk about our work studying them, as well as the title says, how single particle mass measurements have actually allowed us to probe systems and glean biological information that actually wasn't available to us before. So in the first half of this talk, I'll talk about some of our work studying ribosomal particles. In the second half of my talk, I'll talk about our work studying antibodies and how they bind to the SARS-CoV-2 spike protein. So to start off with ribosomes. So ribosomes are quite challenging in that they're both large particles and they're also quite heterogeneous. So here, for example, is just the human ribosome, the ADS particle. Ribosomes differ both by species as well as the organelles that they're from. But the ADS human ribosome is a fairly large protein complex of almost 4 million Daltons. It's comprised of about 70 proteins and 4 ribosomal RNAs. The ribosome itself is actually made of 2 subparticles that are often called the 40S particle, which is the smaller one, and the larger 60S particle. And about 60% of the ribosome mass is actually made up of RNA. So it's a complicated mixture of various proteins and RNA. So in terms of native mass spectrometry, this proves actually a pretty challenging system. However, for the smallest subunit, well, here I show data for the 40S ribosome, native mass spectrometry is actually able to tackle this system quite well. For example, here on the top panel shows a native mass spectrum of this 40S subunit. The spectral resolution is quite excellent. So each of these colored circles represents a series of charge states for one mass. And you can see here that we can measure a mass of the 40S particle at about 1.2 megadaltons. And our resolution is sufficient that we can actually detect a smaller series of peaks on the aside that actually correspond to this 40S subunit that have lost one or two peripheral proteins. So as you can see here, when things work, they work quite well. And the resolution here for the 40S particle is actually sufficient for us, for example, to actually add a ligand. In this case, this is some viral RNA. And we can actually see the mass spectrum change when we add these ligands. And the masses that we measure afterwards match quite well to the masses of the subunit and the addition of the ligand. So this just demonstrates that the strength of native mass spectrometry after the decades or so that we've had to develop it. So while the 40S subunit actually works quite well in terms of measurements, for quite a long time, the full ADS ribosome hasn't been amenable to this analytical approach. So why is that the case? So to discuss that, to help illustrate that, I've prepared this sort of pictorial representation here. And essentially, it boils down to heterogeneity. So as I've demonstrated with the 40S ribosomal particle, native mass spectrometry can perform quite well and be quite a powerful technique as long as you can accurately assign your charge states. So with the resolution that I showed on the previous slide, this wasn't really a problem. However, as samples get more and more heterogeneous, moving to the right across this plot, the performance, or you eventually hit a point where the performance drops quite drastically. So what causes this? Essentially, this is the point where the spectra become too congested or too messy to confidently and accurately infer what your charge states are. So this can occur from quite a few circumstances. Like the hepatitis B example that I showed, you could have too many ligands binding. So there's too many species that are populating the same region of the mass spectrum. You could have, for example, extensive post-translational modifications. Protein glycosylation is quite infamous for causing this. And a third one is actually salt deduction. Generally, with native mass spectrometry, you want to work with fairly low salt concentrations because salt typically broadens out your mass spectra as well and make these quite challenging to analyze. So actually, the ADS particle falls sort of in this right half of the plot with respect to heterogeneity. And that's because the ADS ribosome typically requires pretty high concentrations of divalent magnesium in order to form a stable particle. And this high concentration of magnesium actually has historically prevented mass spectrometrists from measuring its mass. So if we wanted to measure the mass of something like the ADS particle, we need to change our experimental approach somehow. In essence, we need to eliminate this sort of charge assignment reliance on its performance. So there's a couple of ways one could envision doing this. One is that if we had some sort of technique that could directly detect the charge of an ion, so instead of inferring it from its charge state distribution, like I described previously, if there was some technique that could directly detect the charge, that would fix this problem. A second approach that you could think of is that we could circumvent charge reliance altogether. If there was another technique that would allow us to measure the mass of particles without using ion motion, then this would also avoid this issue entirely. So it turns out that these single particle mass methods that I introduced allow us to solve this problem using the methods that I just described. So I'll be talking about two single particle methods today. One is one that we've worked to develop in our group called Orbitrap-based charge detection mass spectrometry. And the right is another single particle technique called mass photometry. So Orbitrap-based charge detection mass spectrometry, it's a variation of what I will now call, for the rest of the talk, conventional native mass spectrometry experiments, in that the experimental setup is quite similar. However, this technique relies on the extremely high sensitivity of modern mass spectrometers. And they're so sensitive now that we're actually able to detect single ions within the detector. So if you take a sample of protein or a protein complex and you dilute it down such a way that you're detecting single ions within your detector, the mass spectrum actually ends up looking something like this on the bottom. So it looks quite noisy and weird. But what I'll notice is that when you look at it, you can actually detect ions. So these are the ions here. And actually each of these ions here as a vertical line is a single ion within the detector. So we can actually begin to start counting ions in this approach. And you can see here also that there's peaks that are double and triple the height. And these correspond to 2 and 3 ions of the same mass to charge. So if you look closely, you'll notice actually that the signal intensity of each of these single ions is slightly different. It turns out that this difference in intensity actually lets us calculate its charge. For the interest of time, I won't go into the math behind that. But essentially, the intensity of each of these single ions is directly proportional to its charge. So within an appropriate calibration curve, if you run your experiments in this single particle regime, you can directly detect the charge of your ions. So we no longer need to infer it based on the charge state distribution, but we can instead directly detect it. And because charge is equal to, the mass is equal to its mass to charge ratio multiplied by its charge, this also gives us a way to directly detect the mass of your analyte. So some of the strengths of this approach, like I said, you can directly measure the charge, so we're no longer relying on charge state inference. In this particular setup, it can be run on commercial mass spec instrumentation that's commercially available. As well, because of this large dilution factor, you actually end up using much less sample than you would in a conventional mass spectrometry experiment. So that's quite powerful. On the other hand, another technique that has helped us a lot is called mass photometry. So mass photometry is another way of measuring the mass of a protein or a protein complex, but it works based off of a fundamentally different physical phenomenon than classical mass spectrometry. Instead of using ion motion, you actually measure the mass of single particles in solution using light. In essence, there's a glass slide, and you can watch your particles land onto the glass slide. And when that happens, you can actually measure the scattering light intensity of your single particle. And it turns out actually the intensity of this scattered light is directly proportional to its mass. So here on the right here, I show an example image from a mass photometry experiment. Here we have two particles landing, and the particle on the right here is brighter, and that's because it has a higher mass than a particle on the left. So once again, with an appropriate calibration, using this technique, you can actually directly measure the mass of a single particle. Some of the strengths of mass photometry is, like I said, because it's not based off of ion motion, it completely circumvents this charge assignment problem. So mass photometry will also let us sort of avoid this issue. And like charge detection mass spectrometry, the instrumentation for mass photometry is commercially available from Refine, so it makes it a little bit more approachable to use. And like the other single particle technique, because we're working in a single particle regime, your sample consumption also ends up being quite low. So to sort of demonstrate what these techniques can do, here I start with the ribosomal 40S particle that I mentioned previously. So on the right-hand side here is the conventional native mass spec data. Here I show the data a little bit differently. So instead of showing the charge distribution, I'm showing what we call the deconvoluted or the zero charge spectrum. And this is basically through processing, we collapse all the peaks into a single real mass peak. So here you can see really that the resolution by conventional native mass spec is quite excellent. We measure a mass of about 1.2 megadaltons. And you can also see smaller peaks that are corresponding to that subunit loss that I described earlier. So this is really the sort of best case scenario sample for conventional native mass spectrometry, in that the sample is fairly homogeneous and the charge state assignment was not a problem. So under these ideal conditions, conventional native mass spectrometry performs extremely well. By comparison, mass photometry and CDMS both still measure the mass of the particle correctly, and actually all three techniques are in excellent agreement with each other. All three techniques measure the same mass, about 1.2 megadaltons, and this sort of supports the validity of these approaches. What may be obvious, though, is that the resolution of these techniques is noticeably lower than that by conventional native mass spec. However, I would like to stress that this is, once again, the sort of best case scenario for conventional native. So the cards are a bit stacked in that regard. Moreover, resolution isn't the only component that matters for an analytical technique. For example, another factor that matters is the analysis time. So actually, the analysis time for these techniques differ quite a bit. Whereas conventional native mass spectrometry can take quite a while, on the order of 5 to 60 minutes. Mass photometry, for example, allows you to collect data fairly rapidly. So this data set took probably less than 5 minutes to acquire. And charge detection mass spec, although it does have higher resolution than mass photometry, the analysis time is also lengthened correspondingly. So there are some balances that you have to play with when deciding which of these techniques suit you. But keeping that in mind, we next wanted to move to a more complex system. So here sort of really highlights the challenges with native mass spectrometry that I described previously. So here I show the data for the full human ADS ribosome, whereas conventional native, you actually can't measure the mass of the ribosome well because of the high magnesium content that I described previously. The single particle techniques actually measure it quite well. So we see a distribution here at around 3.8 megadaltons, and that agrees quite well with the mass of the entire ADS particle. And we also see lower masses here I showed in yellow and blue that correspond to the 60S and the 40S particle. So this just demonstrates that depending on the complexity and the heterogeneity of your system, these single particle methods actually let us measure the masses of protein complexes where historically we haven't been able to with just conventional native mass spectrometry. So there's a few characteristics that we can look at here a bit deeper. One is the mass accuracy. So I haven't shown the data, but measurements were also performed for other ribosomal particles. And for all cases, the masses that we measure with the single particle methods are within 1 or 2% of the true mass. So we think the mass accuracy is quite good. In terms of the calibration, we also find that for measuring these ribosomal particles, it's quite important to have mass standards that are within the mass regime of the particles that you're measuring. However, when you do that, the linearity as a function of mass is quite good. And for charge detection, the main calibration that you have to do is this charge intensity to charge calibration. And we also show that this calibration works quite well across a large mass range as well. So just to summarize this section of the different mass analysis methods, if we look at the performance of the mass measurement technique as a function of how complex a sample is, for example, this is the plot that I've shown previously for conventional native. It works really, really well up until the point where the charge assignment becomes too challenging, and then the performance drops quite substantially. By comparison, these single particle methods, although as of right now, the resolution is quite a bit lower than conventional native, these single particle methods really have a strong strength in that they're less sensitive to the sample heterogeneity. So once your samples become heterogeneous enough, they sort of take over. And in this regime, these single particle methods work extremely well. So in terms of choosing which mass technique is best for you, it really depends on what your sample is and what your research question is. And like I said before, it's not only the resolution that needs to be taken into consideration, but also, for example, how easy it is to do the measurements, for example, the analysis time. So like I mentioned before, mass photometry is probably the most rapid in terms of analysis time, with the trade-off that its resolution is lower. Conversely, with Orbitrap CDMS, its resolution is higher than mass photometry for these heterogeneous samples, but the trade-off is that it takes longer to analyze each sample. And conventional native mass spec is somewhere in the middle of these two techniques with respect to analysis time, with the trade-off that it works quite well as long as your samples are sufficiently homogeneous. So with that in mind, I kind of want to switch gears a bit and sort of talk about an application of these single particle massive approaches and how it let us get insight into a system that previously was not able to be studied. And that's the interaction of full antibodies with the SARS-CoV-2 virus spikes. Yeah, so as a bit of background, I don't think I need to spend too much time talking about this. We've all have been acutely aware of COVID-19, but as a point of nomenclature, so COVID-19 is the disease that's caused by the SARS-CoV-2 virus. And the sort of star in this show has always been the spike protein. So this is the protein that's shown in red here, sitting on the surface of the virus. It's the part of the virus that actually interacts with the receptors in the human body that cause infection. So the spike protein is a homotrimeric protein complex, in that it's comprised of 3 identical copies of the same protein, and it forms a spike-like structure. There's been a lot of talk, especially with this top part of the protein, that's been called the receptor binding domain, or the RBD, because this is the part of the spike protein that actually interacts with the cell receptors. So there's been a lot of interest in this receptor binding domain of the spike. So at least here in the Netherlands, where I am, all of the approved vaccines use this spike as the antigen to elicit an immune response. So because people are quite interested in the immune response to the spike, there's obviously a lot of talk in characterizing the neutralizing antibodies that interact with the spike. So there's a lot of questions one could ask in this regard. For example, where on the spike protein can an antibody bind to? So this is known as the epitope. Moreover, because the spike is trimeric and that there's 3 copies of each of the domains, you may also wonder, well, how many antibodies can possibly bind to one spike trimer? And this is called stoichiometry. So you can imagine that characterizing how and how many of these antibodies bind to a spike is quite important, for example, if you wanted to design a biotherapeutic, but also quite important, for example, in understanding the mechanisms of how certain escape mutants, how those variations can affect an antibody binding. And I think that's quite a topical thing now. So if you're talking about how antibodies interact with a protein, we're really in the realm of structural biology. So in structural biology, I would say there's two main techniques to sort of study these interactions. One is X-ray crystallography, and the other one is electron microscopy. In brief, because X-ray crystallography relies on crystallizing a system you want to study, the antibodies are typically quite large and quite flexible, so this makes this approach quite challenging. So typically, crystallographers will take an antibody, will truncate it. So here, and I show a generic structure of an antibody. The point that I want to make is that an antibody is normally dimeric, but to help the crystallization, you typically chop off one of the arms here, and this is called a Fab arm. So you typically take the antibody, you truncate it, and you often do a similar thing with the spike trimer. It's quite flexible and it's quite difficult to study. So you also truncate it often to just the RBD. And if you add these two things and crystallize them, you can obtain atomic level structures. So X-ray crystallography is really, really excellent for high resolution epitope mapping. And what that means is that you can really see in atomic detail how an antibody can interact with part of the spike protein. A major disadvantage, like I've described, is that you typically have to use quite truncated constructs. They may not fully reflect the behavior of the original full proteins. On the other hand, we have electron microscopy. So this works a bit differently. You don't need to crystallize your proteins, but also because of the sort of the flexibility of the antibodies, it's also quite common using this technique to truncate the antibody in the same way you would in crystallography to generate the Fab. And then you can measure how the Fab interacts with the whole spike trimer. So there's been a lot of work using these techniques since the onset of the pandemic. Electron microscopy has the advantage of being able to probe the full spike, although in general, the resolution is a little bit lower than that of crystallography. And like I mentioned, you typically also have to truncate the antibody to a Fab in order to get structural information. So the sort of purposely provocative statement that we wanted to make is to ask, well, how representative is this quote-unquote Fab-centric approach in structural biology to really understanding how antibodies interact with the spike? And we pointed out that actually it's not the Fabs that are, so the Fabs are the more commonly studied version, but they actually aren't the biologically relevant species. Instead, the biologically relevant species is the full antibody, which I'll call here the IgG for immunoglobulin. So in this pictorial diagram, you can sort of see how there's a few differences between this sort of Fab format as well as this IgG format. For example, you can see that the IgG is quite a bit larger of a molecule than a Fab. So you can imagine that may play a role in how many or how well it can bind. Second of all, the big difference is that the IgG is a dimeric molecule. So there's essentially two copies of the Fab arm for every antibody, whereas in the truncated Fab, there's only one copy. So you can imagine that also may play a role in how these molecules interact with the spike protein. So the question we wanted to ask is, well, do we actually expect that the binding behavior or the binding stoichiometry would be the same between an IgG antibody and a Fab antibody? Keeping in mind that the spike is a trimer, so for every domain, there's 3 copies. So on one hand, you could take the hypothesis that, yeah, you would expect that the IgG and the Fab would both bind the same. I mean, they're both antibodies after all. And in this case, you may imagine that the antibodies bind the spike protein like such, in that you get a seemingly simple binding. Because there's 3 copies of every domain, you expect each antibody to bind the spike with 3 copies. And this would be true in both the Fab as well as the immunoglobulin. On the other hand, you could take the opposite view. Because the IgGs are much larger and because there's 2 arms versus 1, you would say that maybe these antibodies bind a little bit differently. You can imagine, for example, that because of the larger size, the spike actually is able to accommodate all 3 copies of the antibody. So you may end up with less apparent binding than you would expect. Another scenario that you could expect is that because the IgGs have 2 arms, you could actually imagine that an IgG binds the spike protein with both of its arms at the same time, occupying 2 binding sites. So this is something called bivalency, and this would also affect how many antibodies can bind to the spike. You can also imagine the other scenario where you have one antibody that actually binds a site on 2 different spikes. So this would be cross-linking. So the reason why these effects are important, especially these last 2, so these have been called avidity effects is another way to describe them. And avidity is a known effector in terms of the potency and the neutralization capabilities of antibodies. In that bivalent antibodies typically neutralize much more effectively than ones that don't. So these are important characteristics for antibodies. But I would argue that trying to extract these effects when one only studies the Fab instead of the full IgG is quite challenging. So to sort of summarize, Fabs are experimentally quite convenient to use, but we would argue that we really need to look at the full IgG format antibodies to get the complete picture of antibody neutralization. And for that, we need to have an analytical method that can distinguish the binding stoichiometries between an antibody and the spike trimer. So obviously, I'm going to start talking about mass measurements here because mass measurements seem like a pretty obvious way to characterize this. And the reason why it hasn't been easy, and actually this is quite a challenging problem, is that the S trimer is actually extremely heterogeneous. And this is due to its extensive glycosylation. So on the spike protein, there's actually a very large number of glycans. And this has been called the glycan shield, and it's thought to play a role in immune escape. So the spike trimer actually has 66 different glycosylation sites. Each of these sites can have a varying number of sugar molecules on it. So that this actually blows up exponentially in terms of how heterogeneous it is. If you do some back of the envelope calculations, you can estimate actually in one particular spike trimer preparation, you have somewhere on the order of 10 to the power of 75 different mass species. So as you can see, this is quite a challenging system, and you can imagine why conventional mass approaches wouldn't work well. So that's been the case for the spike protein. It's quite challenging to analyze by itself, even on the best of days. So you can then imagine trying to measure the binding of antibodies on top of this is even more challenging. So with conventional native mass spectrometry, it's been thought that this has really been out of reach for conventional native mass spectrometry approaches. However, our ideas was that maybe these single particle techniques, due to their lower sensitivity with respect to heterogeneity, could actually let us measure the mass of these species, and maybe let us to gain some insight into how these biological molecules interact with each other. So that's exactly what we did. So we tried both mass spectrometry and charge detection mass spectrometry on the spike trimer, and it turns out that we can actually measure the mass of this species really quite well. So on the left here is a mass photometry data, and on the right we have the charge detection data. They both agree with each other quite well. We measure a mass of about 475 kilodaltons, and that corresponds to the spike trimer. As well, in both cases, we see smaller species that correspond to the spike monomer as well. So when we compare back to the actual amino acid sequence of the spike construct that we were studying, the predicted sequence mass is only about 385 kilodaltons, which is quite a bit smaller than the 475 that we measured. So what this tells us is actually about 30% of the S trimer mass is just from its glycan shield itself. And this really highlights the sort of structural heterogeneity of these spike proteins. So we were quite happy that we were able to measure the mass for the first time of these heavily glycosylated spike trimers. So what we next wanted to do is see if we could measure antibody binding to it as well. So this was done in collaboration with the virology and immunology groups at the Amsterdam University Medical Center, as well as Ian Wilson at the Scripps Institute. So what I want to make clear here is that in contrast to the structural biology methods that I described previously, where they analyze only the truncated Fabs, here in our measurements, we really wanted to use the full antibody molecule, in other words, the IgG, in order to get a more biologically relevant picture of how this binding occurs. So here on the top, it's just the control. So we can also measure the antibody by itself without any problems. So what we did is we took an antibody, we took the spike, we mixed it, and we just wanted to see if we could detect its binding stoichiometries. And it turns out that we can. So we actually tried quite a few antibodies here. Here I show 3 representative ones. And actually, what was quite surprising to us is that different antibodies that we tried actually bound to the spike with different stoichiometries. So for some of them, this CoVA-125, for example, we actually saw a very weak, if not no binding, in that we didn't really see any new species to show up in the mass histograms. In contrast, with some of these other antibodies, for example, CoVA-231, we actually saw a binding that correspond to one antibody binding. And we can distinguish all these bindings by their different mass shifts in the mass histograms. In other antibodies, for example, with this CoVA-218, we actually saw that there were 2 copies of the antibody that bound the spike. So this test was actually quite surprising. We didn't expect there to be such differences between different antibodies. So what we did with our collaborators is that our collaborators actually generated early on in the pandemic quite an expansive library of monoclonal antibodies. So we took a subset of these as representative antibodies, and we wanted to measure how they bind to the spike. Here we leveraged the sort of ability of mass photometry to sort of rapidly analyze samples. So here are the results of this sort of initial screen using mass photometry. Instead of showing the raw data, here I just show the binding stoichiometry via a heat map. And the point that I just want to make here is that we saw quite a diverse breadth of binding behavior with the antibodies, which once again to us was quite surprising. Moreover, none of the antibodies actually displayed the full binding stoichiometry that we expected. They all tended to have stoichiometries between 0 and 2. So we wanted to figure out why this was the case. The first thing one might think about is that this may strictly be an affinity effect. So we looked at the affinity for all of these antibodies. And for 2 of the least binding antibodies, CoVA-125 and 121, indeed, we think this is an affinity problem in that because of the low concentrations that we use for these single particle measurements, that these antibodies weren't able to bind particularly well. However, affinity didn't explain well the different stoichiometries of all of the other antibodies because the rest had apparent binding constants that are all quite strong and they're all within the same range. So we concluded that there must be a different effect happening here besides affinity that can rationalize all the diverse binding characteristics that we saw. So for the next few in-depth studies, we wanted to focus on 2 antibodies, CoVA-118 and CoVA-215. We chose these because they are both quite potent at neutralizing the spike protein, yet by our data, they seem to possess quite different binding characteristics. CoVA-118 preferred this 1-to-1 stoichiometry, whereas 215 preferred this 2-to-1 stoichiometry. And there was also some potential of these antibodies as biotherapeutics. So we wanted to understand what was actually causing these different binding stoichiometries. So to sort of look into this, we thought it'd be a good idea to actually return to the Fabs that are commonly used in structural biology. They're quite a bit smaller than the IgGs, like I mentioned. So if these lower stoichiometries are because of steric clashes, well, we would be able to tell these. And because these Fabs can't bind bivalently, if we saw lower binding with the Fabs, that would also tell us something. So essentially, we thought that comparing these two formats should help us delineate some of these binding mechanisms. So that's exactly what we did. And to briefly summarize, so we use mass photometry and charge detection mass spectrometry here because of CDMS is higher resolution. But we were able to show that actually for 215, the Fabs actually bind with a higher binding stoichiometry than its IgG. Whereas for 118, the Fab actually shows less binding, or in our hands, actually no binding to the spike, which was quite surprising. So to sort of rationalize what our results were, because for 215, the Fab actually has a higher stoichiometry than its IgG, we concluded that actually the reason why COVID-215 shows a low binding stoichiometry as an IgG is that there's steric clashes that happen in the full antibody that prevent further binding. So this is the steric clash effect, or what we call the anti-cooperativity. On the other hand, 118 shows the opposite behavior in that its Fab has a lower binding stoichiometry than the IgG. So we think this is actually quite strong evidence of bivalent interactions or of avidity interactions, in that with the full antibody, it binds with both of the arms simultaneously, and this is the cause of its strong binding. However, this is removed in this sort of Fab format. So the take-home message that we want to say with this is that the binding of a Fab does not seem to necessarily be reflective of the binding behavior of the real antibody. So to sort of accent this point, we note that for the 215, structural biologists were actually able to get the EM structures of this Fab, and you typically see 3 Fabs bound to one spike, and is consistent with our results. Or actually for the 118, the EM work was unsuccessful, likely because you don't see any binding with the Fab. So to sort of wrap things up, one of the last questions that we had from this is that, well, if both of these antibodies seem to bind less than the 3 copies that you would expect, is it possible that how is it possible that these antibodies are actually still able to neutralize? Because you can imagine if one monomer of the spike is free, it may still be able to interact with a cellular receptor and cause infection. So what we wanted to do is measure the binding of the spike tremor against the human receptor, and that's the ACE2 receptor. So in the absence of the antibody, we can actually measure the binding quite well, and we can see that the spike indeed binds the cellular receptor. However, if we run an experiment in which we pre-incubate the spike with an antibody and then add the ACE2 receptor, we actually see that despite not binding the full complement of 3 antibodies, we still lose all binding of the ACE2 receptor. So despite not binding as many antibodies as we thought it would, these antibodies actually remain fully effective at neutralization and remain fully effective at blocking interaction with the ACE2 receptor. So just to conclude my talk today, I hope that today I've been able to show that single particle mass analyses let us measure complex heterogeneous systems that have historically been outside the scope of other mass analysis techniques, and that we've been able to show that we can measure the mass of intact ribosomes. And in the context of SARS-CoV-2, we've shown that we're able to measure actually the binding of full antibodies, not just the Fabs, with the trimeric spike protein, and that our results indicate that the IgGs may not actually fully recapitulate the binding behaviors, and that despite these lower binding stoichiometries, these antibodies still remain fully effective at neutralization. And the one sort of take-home message that I want to leave today is that really the best method for mass analysis really depends on what your research question is. They each have their advantages and disadvantages, but these techniques are also quite new and they're also quite evolving. So it'll be exciting to see how these techniques mature moving forward. And just to finalize, there's a lot of acknowledgements here with various collaborators, both within the group and out. And if you're interested in the work that I described today, both of them have been published, so they're available if you want to read more about it. On the left, there's a ribosome work that was performed by Sue, Shui, and Sam in our group. And if you're interested in the spike work, it's also published recently in ACS Central Sci, and we have the front cover of this month's issue. So if you're interested in that work, that's where you can find it. And with that, I would like to thank everyone for their attention. I'd be happy to answer any questions.
Thank you so much, Dr. Yin. That was really quite interesting, and I'm sure it sparked a lot of questions among our audience. We already had some questions that came in during your talk, so we can get to them. But just for all of you out there, if you still have a question, please feel free to submit it in the Q&A box on the right side of your screen. Dr. Yin, a first question. You talked about the difference in stoichiometry for Fabs versus full antibodies binding to an antigen. And then I guess I was wondering if you would expect to find a different binding ability of an antibody to a spike protein if you would be able to kind of model the entire viral surface where you have multiple spike proteins present.
Yeah, no, that's a really good question, and we've been discussing that as well. Short answer, yes. Something that I haven't talked about is actually this sort of inter-spike binding, so one antibody binding to two. In our hands, we didn't see a lot of evidence for that. However, on the surface of the virus, you can imagine that these virus spikes are quite close together. So that may sort of increase the local concentration of these spike proteins. So indeed, it could be possible that this binding stoichiometries may be a bit different on the surface of the protein, surface of the virus, as opposed to the constructs that we studied here. But we also think that's a bit dependent on the exact nature of the antibody.
Sure, thank you. Could you explain to us how mass photometry actually works? Do particles of the same size hit the glass at identical times, and is time a factor to measure the mass?
Yeah. So for the interest of time, I didn't describe it that much. Short answer is no. That's not it. In essence, you load samples onto a glass slide, and just through regular Brownian motion, the particles will eventually hit the surface of the slide. So it's not the time that it hits the glass that lets us measure the mass. It's actually the scattering intensity of each particle. Generally speaking, because you're relying on Brownian motion, you can imagine that there could be some differences with when and how long it takes for each particle to hit the slide. But we haven't seen any major effects of that nature, as long as you are fairly short and fast in your acquisition time. So on the order of a minute or two.
Right. And is it possible to measure an interaction over time? Yes. The experimental design is a little bit trickier, but fundamentally there shouldn't be a problem. Yeah. Okay, great. Sorry, questions are coming in quite rapidly. I want to make sure I don't miss out on any. For IgGs that have high monovalent affinity, you would expect that each Fab has a different ability. You would expect each Fab to be able to cross-link to two separate S trimers, which would have the potential to lead to large aggregate species. Do you see this by mass photometry? Oh, yeah, that's a really excellent question. To be honest, going into this, that's kind of what we expected to happen. But we actually saw no evidence of a large aggregate species. What I didn't have time to mention is actually if you look at some of the mass histograms for some of our antibodies, we actually see a small population that's about double the masses that I was describing, and those populations actually correspond to one or more antibodies binding two spike trimers. But we really didn't see any evidence of large aggregate species, at least in our experimental design. We were quite interested in that as well, but we saw no evidence for it, at least with the antibodies that we studied. And there's a question about different kinds of antibodies that asks whether you've looked at other types such as nanobodies with or without Fc fusions, and if there are advantages in respect to avidity compared to classical IgGs. Yeah, that's also a really great
Great question. We're really interested in it because with these nanobodies, especially these fusions, obviously one of the main advantages that they report is this avidity effects. The short answer is we haven't, mostly because of a lack of sample. If whoever asked that question had some interesting nanobodies, we'd be more than happy to look into it. But yeah, we would expect that these assays would be quite sensitive for studying nanobodies and other such things.
There are some similar questions asking about whether you've looked at IgA or Igm binding, and if you can detect that.
Short answer is we haven't. So all of the IgGs that we study here were monoclonals. We don't see why other antibody formats like IgAs wouldn't work, but we haven't performed those measurements.
Okay. Does the molecular shape of a molecule, for instance, whether it's fibrillar or globular, would that impact mass photometry?
Yeah, that's also a great question. It's something that we're also quite interested in. The short answer is that we've seen no major effects yet. We've seen no major effects yet with respect to the 3D shape.
Okay, great. This is a follow-up question about trying to look at interactions with multiple spike proteins. Would it be possible to kind of coat different spike trimers, like 2, for instance, on a surface, and then look at the interaction with mass photometry?
It's a bit tricky. I mean, that begins to approach basically the assay format of basically biolayer interferometry. I know there's been some work by other labs with functionalizing their glass surfaces. It would perhaps be possible, but if I can guess what the intent of that question was, there may be better ways to probe that. For example, if you just wanted to fuse two spike trimers with a linker, for example, that would probably more clearly address that research question.
Okay. Could you comment on the size range and limitation of the different methods that you talked about today?
Great. Yeah. So for charge detection mass spectrometry, we're limited by charge sensitivity. For mass photometry, we're influenced by basically shot noise. For both of the techniques, it's a rough ballpark. 50 kilodaltons is probably the lower limit, up to, I would say, 5 and 10 megadaltons. But this is really, because these techniques are still quite young, there's still a lot of work to be done in terms of pushing the upper limit. But as a sort of rough, I would say it works really, really well between 50 kilodaltons and say 1 megadalton.
Okay. Could you try to model the interaction with only the part of human ACE2 that interacts with the RBD rather than the entire molecule?
Yeah. So I didn't mention explicitly, but actually the ACE2 construct that we use, I mean, yeah, so you could truncate the ACE2 domain. We were kind of hesitant on doing that for the same arguments of the IgG versus the Fab. So for us, we studied the dimeric. We had a dimeric construct of the H2 ectodomain, but I don't see any reason why you couldn't truncate it, for example, to just the monomeric ACE2, particularly just where it binds with the spike. I don't see why those experiments couldn't be performed.
Great. Here's a question about, at some point you mentioned that about 30% of the spike protein was glycosylated based on your studies. It's a question about controls for that experiment. Have you tried to deglycosylate the spike trimer and then repeat your experiment?
Yeah, that's a good question. So I'll first answer that by saying we actually compared the glycam acids that we got with bottom-up glycoproteomic data, so the sort of gold standard method, and those numbers actually match up quite well. We haven't tried deglycosylating the spike trimer, mostly because those deglycosylated constructs tend to be quite unstable. So experimentally, that's quite hard to do. But we don't really expect any biases because we've measured many other proteins within that mass range. So we don't think mass calibration's an issue or as such. But I hope that answers the question.
I think it does. It sounds to me like it does. I guess I was wondering whether, based on your experience, if you have a highly glycosylated protein, would you recommend scientists to use mass photometry to study that protein, or what is your experience?
Yeah, so basically our thoughts with that are like I said, if you're interested in mass measurements, we really think these single particle methods are the way to go. Because when your proteins are heavily glycosylated, the sort of conventional mass analysis approaches really don't work very well. So we think these single particle things are really essentially tailor-made for those types of systems.
Great, thank you so much. I think we have time for just one more question, and it's a question that asks you, in light of the research that you've done and the findings that you've made so far, what would be the next question that you're most interested in pursuing?
That's a great question. I mean, without revealing my hand too much, I mean, we're really interested in how antibodies and other immunogenic molecules, like other antibody formats, for example, interact with these viral proteins. We're quite interested in basically pushing this mass envelope higher and higher. So here I've shown data for just the spike trimer. We're interested in sort of larger, maybe more realistic constructs that maybe have more than one spike, that maybe a pseudovirus or something. That's kind of what our dreams are, at least some of it.
Perfect. Thank you so much for joining us and giving this awesome presentation. Unfortunately, that's all the time we have for today. There are still quite a few questions that we didn't get to today. And on top of that, if you have any other questions, you can definitely reach out to Dr. Yin directly, and you can see his contact information shown on the screen right now. As a reminder for all of you, this webinar will be archived on the Scientist website, and you will receive an email notifying you when the on-demand webinar is available. I would definitely like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On the behalf of the Scientist, I would also like to thank our speaker, Dr. Victor Yin, as well as our sponsor, Refine, for today's webinar. Thank you, everyone, for tuning in, and we hope to see you again next time. Have a great day.